I don't know. Let's find out. I don't know. Just saw 12 something. Anyways. Come on, come on. Position. A right shift and more effect. MPCO2. Acidic environment. D. 2,3-DPG. Exercise and increase in temp. Yeah. Now it's also from the variation range of something known as. Do something known as. There's something known as action potentials, neurotransmitters, Newton wire. Negative coverages and it doesn't have a lower body affinity. Lower affinity. Right shift indicates a right shift indicates a decrease in global affinity from oxygen. The axon is continuous, and technique that's called around here. Instead of oxygen, it's jumps. I'm gonna jump. Heterozygous. Adaptive, specific response time is hours to days. Very slow. It's called a sarcodon. VF equals VO plus 218. No, it's not. VF equals VO plus 18. So there's I think this is okay. Let's just see beyond cycles. Beyond. Go above and beyond. Intricacies of hemoglobin reveals the involvement in maintaining enzymatic processes, orchestrating cellular respiration. What was the other one? I forgot the first one. You got me. Homeostasis. That's homeostasis. Homeostasis. Equilibrium of reaction deals with the concentration values of reactant and product, which there is no net difference. Well, no net change. Okay, well. Alright. Macrophages, dendritic cells, macrophages, dendritic cells, natural killer cells, monocytes, mast cells, and then neutrophils, basophils, eosinophils, Come here, Grandpa. Granulocytes. You, Grandpa. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4. 1, 2, 3, 4, 5, 6, 7, 8. Yep. Let's go. Good job. This involves one division resulting in two genetically daughter cells, two sets producing four genetically diverse daughter cells. Cells releasing those various weights, interaction differentiation, autocrine to self, justicrine to nearby and adjacent cells, really close. I'm not sure if that's S or not. Paracrine to cells that are further away, endocrine to cells that are very, very far away. That's endocrine through the bloodstream. Around but not attached. Because there are components of the neuron. So that stuff is like, duh. Introns serve in introns. Alright, let's see if I can get the wording exact. I don't know if I can. Introns serve. Let's say in principles, but no. Introns serve. I don't remember the exact words. Anyways, let's see. Introns serve in organizational splicing and epic. Oh, introns serve various roles, such as organizational, splicing, and epigenetic roles in gene expression. Oh, that's kind of close, actually. Oh, actually, it's pretty close. It's not as bad as I thought. Neurons has the messages that we need to think and carry out action. Neurons also responsible for their own communication to the neurons. These neurons send signals to muscle gland cells, muscle cells, or glands, muscle glands, muscles. Don't move, muscles. Benefits of neurons that may or may not be violated. So it goes synaptic vesicles, which are saying neurotransmitters. And just go ahead and dump some transporter into the synaptic. Into the postsynaptic. Into the cleft cavity. What is it called? Cleft area? Cleft cavity? Synapse. It's called a synapse. The activated alpha subunit inside of the surface. There are ways. Microglia come in the same lineage of precursor cells. It's too easy. Dendritic, these branch-like parts are only on the receiving end of the message. They do not send the message or send neurotransmitters. Because they listen, right? They listen. Because they listen. Do they listen or do they talk? No, they listen. Axons talk. Dendritic dendrites listen. So they receive the messages. Yeah, something like that. The activated alpha subunit or the beta gamma complex. Why is that still here? It's between motor neurons and sensory neurons, strictly within your CNS. Sympathetic nervous system is active during. Sympathetic nervous system is active during. Fight or flight situations. Let's go, buddy. We got all day. CNS can process yo. Brain sparkle. Proteins. Ion. Well, receptors and protein channels. I'm a protein. Pressure gradients. Well, we have diastole and systole. Diastole, we have let's just go with systole first. Systole, we have blood rushing in, pulling at the ventricles, relax at the ventricles. So we have the pulmonary valves closed. We'd also have the AV open because they just open to have the blood. AV valves open to have the blood pull into the ventricular cavities, and then so that's lub, right? And diastole is basically the opposite. Yep. Elicit a response, transmitting signal from outside the cell inside the cell, right? Activate or inhibit other proteins. Myelin, salt-door conduction. Wait, no, myelin sheaths. Myelination. Glial cells supporting difference. Let's go. Glial cells are any cells that help neurons do their job, as we refer to them as helper cells. Which they are what they do. Nervous system branches out from a vast. Oh, I think I have them here, right? Wait. Rock side is on. I should have gone down. Regions, nervous system branches out from a vast CNS and PNS. And no, I'm not saying penis. That probably sounds like it. Cell body is a central part of a neuron. It helps to make sure that the neuron is healthy, and also helps to receive signal, right? Transmit signal. Processing. I'll look at another one. Cellular response within the cell. Well, yeah. Axon. Send messages out to other neurons through electrical or chemical synapses through neurons, other neurotransmitters. So, yeah. Away from the cell body. Hippocampus, amygdala, thalamus, hypothalamus, amygdala. A neuron and I got some more. A neuron and the what? A neuron and What's inactive left? No. Target sale. Oh, right. It's just trying to tell us this can be between different cell types. Right? Maybe. Um, so, so given the probabilities of two independent events, and if you want to find out the likelihood that the likelihood of both events occurring, you're going to multiply those independent probabilities together. Let's go. Let's go. Spot on, dude. Spot on. Physiological responses to the. That's definitely the sympathetic, parasympathetic. Deal with motor function, regulation of emotion, and memory. Oh, I guess never mind about the motor functions. It's just emotion and memory. Mood and emotion. Sorry, mood and memories. Um, I still have a little bit more for this. Yep, maybe, but at least you brought it down by a lot. I could do this, the other parts a little bit like in the bathroom or whatever. I don't know. I could like fill it in. But the bulk of it was this, which I fell behind. Anyways, target cell commonly seen with just any cell that receiving any of the synapse, glands, or muscle cells. Acetylcholine, epinephrine, norepinephrine. Um, yeah, just those three pretty much. Understand high blood osmolarity or the blood volume. Light, sound, touch, anything sensory basically. They basically just help support and provide insulation for those neurons, right? Schwann cells for the PNS and um, oligodendrocytes for the CNS. Spit on that one. Yeah. Point cells, cells, non-germline cells, somatic cells, somatic cells. And the nucleus that contains two sets. Um, well, in a set, it contains two chromosomes, a pair of each chromosome. Set. Heterozygous, two different allele genotypes, well, two different allele genotypes. They gave little a genetic, different genetic information, two different alleles, dominance, phenotype, the physical trait expression of a particular allele, or what is expressed. Does it have to be that? No. Myelination is the basis for something called central connection. Dominant allele, an allele that is dominant altogether, one that doesn't need both alleles to be present for it to be phenotypically expressed. It just needs one. Parasympathetic, more than relaxed situations. Was it Russian Digest? Star-like cells, those from the BBB, astrocytes, they create the physical barrier. Cell body, central part of the neuron. Yeah, nucleus, yeah. Okay, come on. Motor neurons, sensor neurons, interneurons. Oh man, 166. Neurotransmitter is a messaging signal that is released by uh, these released by uh, the presynaptic cleft. Presynaptic cleft. And then we shoot at a target cell. Send a message. Right. I don't know these neurons. Carries messages. Yeah, I'll show you this one. Trying to send it. I'll do one more. I think the one is that um, elusive traits are made independent of one another, essentially, right? Or inhibit independently. Yeah, kind of the same. Yeah, I thought. The nervous system branches out from the vast overview, large anatomical regions involved, central nervous system, penis, to stimuli, to stimuli. Um, uh, okay, physiological responses, parasympathetic, sympathetic. Sympathetic includes increased heart rate, blood pressure, dilate pupils, decreased auditory glands, so the decreased saliva. I think just concerns for that. Um, and there's another one. It's like just decreased enzymes for digestion, I guess overall. Yeah. Um, I don't know. I think I saw it would release um, relax bladder, relax the bladder, but I'm not sure about that one. Penis liver divided to nerves and urinary muscles. Oh, you. Motor. Oh wait, motor sensory neuron, somatic nervous system. Oh, your autonomic nervous system. Either or and. So if we have um, two probabilities, and we have want to find out the likelihood of one or the other occurring, we're going to multiply those. I'm sorry, we're going to add those probabilities together. Individual amino acids undergo a condensation reaction and fuse to form peptides, which are held together by something called the bond. Includes the nodal cord, acceleration, Acceleration doesn't just imply speeding up, acceleration is also the time encompassing alternations in both speed and direction. Sports that encompass forms of mental synchrony, they form during link dental synchrony, they are formed during dun, 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 dun. Linked synchrony, they are formed during I am bane. Batman. Anyways, they are formed during chromosomal replication, so during the S phase, right? Cytotoxic T cell is marked by CD8 plus on their surface. Kill cancer, by the way. This measure is summarized contributing to the segmented structures of the vertebral column. Not gonna get into this time, buddy. Fetal hemoglobin, surface anemia, beta thalassemias, so reproduction beta HB and HBV virus. Equation is fundamental. VF is equal to VO. VF equal to VO plus AT. Well, hemoglobin is one of those things with dividing against the ocean with energy. You want to see beyond, huh? Go beyond the stars. Maintaining homeostasis. I'm not sure, but I'm going to say facilitating enzymatic processes and orchestrating cellular respiration. That's fine. Cell body is a central part of a neuron. It also helps process the information, process the incoming messages. Target cell is target cell. Any cell there's well, I guess maybe not. Well, maybe cells are designated as a postsynaptic. Well, excuse me, on the postsynaptic side, maybe. Anyways, commonly seen. Postsynaptic side of the synapse receives the message. Muscles or glands? I want to say muscles or glands. Ooh, let's go. Okay, yeah, that was kind of close. I guess. Something. That's fine. Neuron and the target cell. Cold. What are they called, buddy? What are they called? Myelin sheaths and a myelinated axon are cold. Nodes around the ear. Oh, 23? Oh, maybe I should have done 23. That's fine. Okay, what do we got? Mouth to the rectum, endoderm, trachea, endocrine glands, gallbladder, liver, pancreas, bladder, urethra. I want to say. Oh, we already said urethra. Let me say urinary bladder. What do we got? We said GI tract. Well, we didn't say pancreas. Oh, we said pancreas. Dang. Shoot. Gallbladder. Urinary bladder. Oh, we didn't see the glands yet. We got thyroid, parathyroid. Oh, we got lungs. We got lungs. I don't know why I forget lungs. But sure. I always forget lungs, huh? Non-competitive game does not move. That's known as the same. Let's do good on that. Three molecules is kind of side of the DNA strand. We're just cold. Okay, so we got the mRNA, which is on the non-coding template strand, and it's almost identical, would be, we would say, uh, to the coding strand, right? Coding strand or anti-sense strand. There we go. Boom, baby. Boom. Lock that in. Locked in. Objects persist in a state of rest or motion. Example. We have inertia. Example. We have an object. We have an object with bound sources. So we have gravitational force, force of friction, depending on if they're like, yeah, a couple other things. So we will need an external unbalanced force in order to move it, and the force would be greater than the balanced force in order to actually move the object. Let's be unbalanced. Yeah. Inertia and momentum. Yes, sir. Thank you. Clear proteins. Glucose transporters, or I'm not sure, buddy. Oh, what? Okay. Alrighty. Imagine a protein as a hallway with two doors. As a hallway with two doors. In a carrier protein. Wait. Okay, are we inside the hallway? Maybe. Okay. Only one door is open. That's interesting. Path. Alright, transport. Okay, I guess. Sure. Autosomal. Autosomal. Autosomal versus self. Self in what way? Self in terms of cancer, autosomal, self in terms of immune system, grafting. Thinking about grafting. Autosomal grafts. No, autosomal allograft. I'm blanking out, dude. Autosomal. Well, no. How about allelograft? Allelograft? No, I'm pronouncing that really bad. Allograft. One individual to another. Seems to be self, right? I'm not sure. That's way off. Okay. 122, accept the sex chromosomes. Messed that one up. Okay, pair up. And change in cancer meiosis. Second week, position, congratulations for the muscle. Mesoderm. Sorry, it should be endoderm. Well, I don't know if what order it happens. I should find out actually. Yeah. Anyways, ectoderm, mesoderm, ectoderm. But actually, I want to find out. That's why I want to review embryology with Andrew. There's some questions I got. There's some gaps in his knowledge. I didn't even type in a question. Oh man. Whatever. We won't wait for it. fertilization is an event that changes the chromosome count, starting with, wait. Fertilization is an event that changes the chromosome count. 23 chromosomes, ending with, okay, 46, right? Yeah. Because you just got fertilized, son. Are part of and can act as APCs. Hold on. Okay. What are we talking about? Part of blank. Beta macrophages to dendritic cells are part of. No, because beta is a. Beta cells are part of the adaptive immunity, right? Humoral. That's right. That's right. Is that true though? What I just said? No, right? If that's a separation, it's humoral. Or no? Well, it is. Well, I think it's like a derivation. Whatever. Continuous with the. It's continuous with the. Uh, 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 with the nuclear envelope. And the smooth. I didn't know that. I guess so. Yeah, well, transported to the Golgi. Threshold potential is reached. When the threshold potential is reached, we got Na plus channels open, causing rapid influx of Na channels, anions into the neuron, polarization, final charge to these Na plus channels are voltage guided, meaning that threshold potential is reached. Mesoderm, we got muscles, blood, muscles, bone, blood vessels, connective tissue. So it's not lungs. Is it kidneys? Let's go. This is like freezing on me or something. Yeah, let me see. Warren, CD. Wait. Yeah, CD plus, and then MHC through CD4 plus. Key member potential numbers are negative 7V threshold. Peak actual potential around plus 40V. And the top state HB is. Okay. 10 minutes. I gotta take my alcohol. In the top stage, HB is not bound as low affinity. Hollow cell mass, and with a cavity called blasto. Wait, Blastocell? No, Blastocell and it contains intersomas. After this one is the, I think, um, I'm not sure. Anyways, let's just, yeah, that's fine. I forgot what the order is, but we can go over it and we go in embryology a little bit. Hopefully, Yusuf kind of has some good knowledge on it. Conformational change, initial state of the innate pump. Well, I'd say it's closed, right? And then, yeah, let's see what it says. This is a new one in this. Really, that's the initial state. Oh, right, because I think it constantly goes, right? I mean, if I remember correctly, I think I noticed it constantly goes. And this one didn't work. Okay, so pump open the inside of cell, align the binding of 3 Na plus ions. Initially, it will cascade produced in the pancreas. Insulin is has isolated Langerhans, and it's the cell type, or it's this islets, islet. I think insulin is islet beta, and glucagon is islet alpha. Is that right? Oh, so the endoderm is formed first, then then ectoderm. That's good. Cool. Thank you. Pancreas islets of Langerhans. The islet of Langerhans. What did I say? I said glucagon is alpha and insulin is beta. Let's go. This includes the GI tract of the mouth, the rectum, the trachea, the bladder, pancreas? Pancreas. Oh, I said pancreas. Oh, I think this one's lungs. Yes. Initial state open. 3K. Oh, 3Na plus. 3Na plus go out. 2K plus go in. And so it's Oh, but I think the best thing to remember is this: pumped open to the inside of the cell. The pump is open to the inside. Oh, so much. You can change to our other one. I gotta take a shower too. Pump's open to the inside of the cell. Only binds to competitive. Uncompetitive only binds to the ES, right? ES, yeah, ES, yes, ES complex. Produce. Germ cells start as diploid cells, reproduce blood gametes through meiosis. Um, I'm not sure. Oh, haploid. Darn. active cis transport. Yeah, I don't think. Okay, I mean that's fine. Wild type, we have as positive or false positive. Usually used when we're trying to determine um, well, some sort of experiment. So we have wild type versus control versus uh, um, experiment. Well, right? I mean, the mutations on them, phenotypes on them. Um, the alleles represented as assumed to be homozygous dominant. Okay. Refers to the chromosomes that are not sex determinant. So the 22 other 22 sets of 22 sets of chromosomes. Right. Okay, so two genes of the ones facilitate diffusion. HPV and HPV variants, HPV parts, HPV parts. 4 voltage is characterized by the all or nothing firing of a neuron. Yes. It's manufactured with changing the pH or increasing temperature. Usually, the protein correct. Coming down. Most expressed phenotype. Codominance. Codominance. These cells ingest the pathogen and present antigen 2A. T helper cell, usually T helper cell um, uh, that has a CD4 plus on it, and active is equal to a large number of antibodies between to the plasma cell or a memory B cell. Yes, sir. Yes, ma'am. Function is to modify, distribute a protein, and this is in eukaryotes only. Um, and then I'll go ahead and send it out. It needs to, right? Shipping them out. Gonna get shipped out. Gonna get shipped out like I'm getting shipped out. Air Force. Fight, flight. Wait, fight, flight. Oh, Air Force strong. But I'm not sure. I don't know the whole model thing yet. I'll get there. Displacement refers to the change in position of an object. Change in yeah. Displacement from the starting position. No, um, the position ended compared to the position it started. Or vice versa, I guess it doesn't matter. Okay. Vector quantity. Yeah. B cells are probably more immunity. Genes are genes are genes are uh, segments of DNA that code for proteins, or yeah, segments of DNA code for proteins. That particular genes are specific segments of DNA that well, it depends. Like, if you're on the genes and genetics, I guess it'd be code for specific traits potentially. Code traits cover routines, passing paradigms. Yeah, that's fine. What's this with the changes of time? Moving at 10 meters, 10 meters, 10 meters per second. This and wait, I want to say inducements. Okay, yeah, inducement. Good, good. Pancreas. Yeah, there's something pancreas, gallbladder, urinary bladder. Also have the lungs and the urethra. No, that's not there. Macrophage and dendritic cells are. Oh yes, yeah. A part of the innate immune system. Breaking down. We break down reactive oxygen species and well, that's what I know most of them. Breaking down reactive oxygen species, I do a host of other things, but they also generate some intermediate reactive oxygen species, like radical radicalized hydroxyl, hydroperoxide, and I think radicalized O2. Mm. Fatty acids, amino acids, and detoxify chemicals. F equals M times A. For B cells, the key points are under B cells. And development of the nervous system. 6 chromosomes. X, XX for female, XY for male. Comparative or comparative, not comparative. Relevance, pertinent ions. And also it's contributing concepts like relevance, concepts like electrochemical gradients, resting memory potential, and neuron voltage. Okay, resting memory potential and neuron voltage. Okay, I think I'm okay with that. Okay, so fatty acids, amino acids, and detoxify certain chemicals. Maybe let's do this in aminos and genotype. Homozygous. Homozygous dominant. Homozygous recessive. In the relaxed state, this species malfunction has high affinity O2. Most thought that the genes created a shift off to undergo um, PTM. No, yeah, PTM. Including protein folding. Protein folding is happening with the chaperone proteins, right? This is the peak energy curve, which must be a call for reaction to proceed forward. Transition state. Insulin are glucocorticoids are um, I'm pretty sure your steroids. You have to be right. Well, you don't have to. I think so. Ah, are you peptides? Seriously? Seriously? Oh, they're peptides. Oh, okay. Never mind. Got it mixed up. Most of them are peptides anyways. I think most of the major ones. Go against it. It's open inside the cell. Well, yeah, it's allowed 3 NA plus and 2 K plus. Allowing the binding of 3 NA plus ions. But I think it just means like its initial, its initial conformational change, which is just it's already binded to 3 Na plus ions, right? Like I think if we probably see a picture, you're gonna see with the actual structure that it has 3 inside already. Like, well, maybe not like initially, but in order for it to actually begin, right? I think so. I'm probably gonna see that. Yeah. Facing, I'm not sure. Well, I guess it says it's open on the inside. So I guess it's open inside, right? Open inside, and then once we get well, let's see. Any pump changes shape. It changes shape and requires energy. I'll look into the change later on. Actually, I could just type it in. Explain to me the conformational changes. And number them as as the process proceeds. Yeah. Something like that, right? Use deploys. Oh. Man. I'm tired. DNA libraries bind to molecules together. Yeah, there we go. Okay, I have the conformational change here. Yeah, so the edges of the repair DNA. Oh, okay, this is during NER. Yup. Produce some sort of, well, I guess produce some sort of intracellular response, right? Generate secondary messengers. Okay. Oh, yeah, okay. GPCRs. That's fine. There we go. With the conformational sign right here. Pump is open to the intracellular side. 3 ions from inside the cell bind to the pump. Okay, then ATP hydrolysis and phosphorylation. ATP is hydrolyzed, transferred the phosphate to the pump. Yes, pump gets phosphorylated. So yeah, so ATP is hydrolyzed, transfer the phosphate to the pump, change conformation to the outside. We release the Na plus, taking the 2Ks, and then phosphate gets removed. Reverse, we flip it, flip, and then we release the K inside the cell. There we go. That's not that bad. I gotta take my. I'll be right back. I got a lot. I shouldn't let this happen again. Innate immune system. Oh no, adaptive immune system. Yeah. Sync it. Yeah, I brought down a lot. I think the rest of probably just take me like maybe an hour. I'm not sure. Did quite the many cards today, I guess, though. Relaxed states, relaxed. Nasty. Nasty, nasty. Alright, guys. I think we just probably call it here. Let's call it here. Finishes tomorrow morning. And then we have wait. I need to add the physics. Oh my god. Physics and math. Yeah, circle. The venturi tube. Yeah, the venturi tube. Um, I don't like the way this one's set up, to be honest. Oh, actually, wait. Newtonian laws. Do-do-do-do-do-do-do. Do-do-do-do-do-do-do. Oh, not circus. No, no, no. No, sir. No, sirree. Electrostatics. No. Energy. Energy. Mm-mm-mm-mm. Yes. Come here, energy. Hooke's law. Maybe. Fluids. Fluids. Yes. Boom. Gases. Why is there some here again? Light. Fluids. Ah. Energy. Fluids. Gases. Is that really all for gases? Oh, and there's only one. Sure. Okay. Yup. Yup. Wait. Oxide is on. Look at this spacing. Absolute pressure. Yup. Get in there. I think so. Yup. Yup. Specific gravity, substance. Rate of volume, probably. Anything to do with fluids, I must include. Specific gravity, fluids in motion. Fluids in motion. Gases. Gas. Yup. Yup. Spring potential energy. Energy. Oh, but gas pressure. No, seriously? Okay. Hooks law. Something's wrong with it. Or maybe not. Fluids in motion. Pulsarily flow. Pulsarily. No. Flow rate. I guess not. I don't know. Working energy. Which deck is this? Oh. Thermodynamics. No, I'm not there yet. Fluids. And that's it, yeah. Okay, hold on. This one, it didn't really properly work, which is a shame. Just wanted to know what I was trying to say. I don't know what it was saying. You know? What's the other one though? I don't know. Oh, working energy. Psh, come on. And the heart is a pump. I do gas law. Yes. Circuits. Thermodynamics. Oh, why is it doing this? Oh, never mind. Go back. Oh, what? I don't like this. I don't like this. Oh, can you stop? I really don't. Sort the field, maybe? No, I don't know. Wait, what is happening? Let me close it out. It's being all weird. Maybe it just needs more space. Maybe. What if I type in physics? Physics. That's fine. This doesn't work. There's only a little bit on here anyways. Yes. We're getting there. Yup. We're getting there. Yup. Alright guys, I'm gonna call here. Oh, there you go. Now we have some physics. That's okay. I'll see you guys tomorrow. Peace.